Hello and welcome to this presentation. Today we will talk to you about the selection of yeast and fermentation conditions for the production of Sauvignon Blanc. I am Flesch from Fermentis and I will present with Marie-Charlotte Colosio from Institut Français de la Vigne et du Vin on this collaborative project. So today's agenda, I will start with talking about the impact of yeast on Sauvignon Blanc's aromatic profile, both fermentative impact, thiols, and we'll talk about some specificities. Then Marie-Charlotte will talk about the process of the selection and screening of yeast for Sauvignon Blanc. And then I will come back to present you a case study trying to show the influence of yeast strain and fermentation parameters on Sauvignon Blanc. And finally, present you some data from validation in the field of the selected strain. So Sauvignon Blanc, of course, we know is one of the most widespread aromatic variety through the world. Actually, it's number 11th most planted variety in the world. It is a strong variety in France, but more specifically an emblematic varietal for the South Hemisphere. In Northern America, it's very important, and especially in California and Washington. So what defines Sauvignon Blanc's aromas? First of all, strong varietal flavors. In the first category of the varietal flavor, you're going to have the pyrazine that kind of confers some green, asparagus, bell pepper type of aromas. Here, the main influence parameter is going to be the date of harvest. Then you're going to have the thiols with here 2 molecules, the 4-MMP, kind of promoting box tree, blackcurrant type of aromas, and the 3-MH, more on the citrus type of thiols, grapefruit, and passion fruit. But the strong varietal flavors are balanced with fermentative flavors that do not come directly from the grape, but from the fermentation and the yeast itself. And in this category, the first family is going to be the higher alcohols. The most emblematic one is going to be the 2-phenylethanol, conferring aromas and flavors of rose, typically. Then you're going to have a first category of esters called the linear chain ethyl esters, going from the ethyl butanoate to the ethyl decanoate, and they tend to confer overall complex fruitiness to the wine. Finally, we have another category of esters that are very aromatic, the acetate esters, with the most well-known and important one being isoamyl acetate that confers banana, strawberry, candy aromas to the wine. And finally, a molecule and aroma that is kind of between strong varietal flavor and fermentative flavor is going to be the 3-MHA. So the precursor is going to be the 3-MH, and it's going to be esterified in an acetate esters. So that's why it is both a fermentative and varietal aromas. And it is associated with more tropical fruit, tropical thiols, such as passion fruit. The yeast is going to have an impact on, of course, the fermentative flavors, but also on all these thiols that we talked about, 4-MMP, 3-MH, and 3-MHA, and we'll see that next. So the choice of yeast is very important for Sauvignon Blanc. So here we have this in a little bit more detailed way. We have a yeast cell here. Of course, we use yeast in order to ferment the glucose and the fructose into ethanol. In the process, the yeast is producing fatty acids that are going to be important precursors in aromas. The yeast is also assimilating some nitrogen sources, so mainly amino acids and ammonium from the must. In the process of using the amino acid, the yeast is producing a byproduct that is very aromatic, that are the higher alcohols. From there, the higher alcohols can be esterified with acetyl-CoA into acetate esters that we mentioned earlier. So an example, for example, if you want to follow an amino acids, if you start with the leucine here, the leucine is going to give the higher alcohol isoamyl alcohol, and the isoamyl alcohol is going to be esterified in isoamyl acetate. Both of these molecules providing candy and banana flavors to the wine. Finally, the last category of ester, the ethyl esters, are going to come from the esterification of the ethanol and the fatty acid acyl-CoA, and they're going to bring overall fruitiness. So these are all the main fermentative aromas. We said the yeast has an impact on the varietal aromas. Now, these aromas can be expressed or non-expressed. In the must, you're going to have some thiol precursors. So these precursors can be cysteinated, which means linked to a cysteine, or glutathionated, linked to a glutathione. And they're going to be imported within the cell, where through different reaction, the yeast is going to release the volatile thiols, the 4-MMP, and the 3-MH. Then the 3-MH can be esterified in 3-MHA, and here you have the production of these three main thiols. So just focus on the higher alcohol and esters. Here it's exactly the reaction that I was presenting earlier. So the ethyl ester, the acetate ester, and the higher alcohol are going to be the main compounds produced during fermentation that are responsible for the fruity and the floral aroma. What I wanted to highlight here is that it is a highly regulated process. So here you have all the main genes that are responsible for coding for the different enzymes and transporters that the yeast cell needs in order to produce these molecules. So it is highly regulated, and it is depending not only on the strains, but also on the conditions. So for example, we know that temperature is going to have a positive effect on the gene ATF2 in order to produce acetate esters. But we also know that overall these acetate esters tend to evaporate very easily with high temperature. So these are examples of things that we know on this regulation. Looking at the thiols more specifically, so these are the reactions that I presented earlier. So the 4-MMP, the 3-MH, and the 3-MHA are going to be the main compounds released specifically more at the beginning of the fermentation, and they're responsible for tropical for the 3-MHA, more citrus for the 3-MH, or greenness more typically for the 4-MMP. Once again, it is a highly regulated process, and here we highlighted the most important genes that are responsible to code for different proteins. Because it is highly regulated, the conversion rate from the precursors into the volatile compounds is actually pretty low. So here you have the main genes that are involved are going to be the ERC7, that is going to code for the enzyme responsible to release the 4-MMP, or for example, the STR3 to release the 3-MH. One of the main regulation system in place here is called the nitrogen catabolic repression, that is going to regulate different genes here, all the ones in purple, so ERC7, GAP1, and OPT1. So looking more specifically at the ERC7 gene, if you look at the genetic level, you're going to have one copy of this gene on both alleles. One version of this gene could be the ERC7L, standing for long. If that's the case, this is going to be transcribed into a long RNA, and this is going to be translated at a protein enzyme level into a carbon sulfur lyase that is going to be active. So when it's active, what it means is that it's going to be able to release the 4-MMP from the precursors. Now, if you have a version of the gene that is S, ERC7S for short, meaning it's shorter by 38 bases compared to the long version, it's going to be transcribed into a short RNA, and it's going to be translated into an inactive enzyme at the protein level, which means that it won't be able to release the 4-MMP and act on this precursor. So yeast have three possible genotypes. They can have an ERC7 long on both alleles. They can be short on both alleles, or one of each. Most wine yeast have too short version of these genes, which means they won't be able to act on these precursors. And if they have long or long short, they will be able to, in some proportion, release the 4-MMP. Something else that we know is that the ERC7 gene is downregulated by low temperature. Okay, so now let's talk about the Ncr regulation system. So you have available nitrogen into your mast coming from the composition of the mast itself or potential additions. The yeast is going to use this to produce biomass and to complete fermentation. So it's going to have an impact on the speed of your fermentation and on its completion. It will also have an impact on the production of flavor compounds and other metabolites. Now, yeast will always prefer nitrogen sources that allow their best growth. And the way this is done is by different regulation systems that are in place. The two main ones we're going to talk about today are the nitrogen catabolic repression, NCR, I will call it NCR from now on, regulation system, and the SPS, specific permease system, sensor regulatory mechanism. So if you look at these two systems at a genetic and phenotypic level now, here you have the sequential consumption of the nitrogen sources. And you have here a yeast cell. So you can see that in the first place, the amino acids that are in orange are going to be transported through permeases that are controlled by the SPS system. So they're going to be transported in a specific order. And then secondly, you can have the ammonium and a group of more general amino acids in green that are then transported through general carriers that are downregulated by the NCR. So once again, this is going to happen in a specific order, usually when the concentration of the orange amino acids, especially the glutamate, is low. So specifically, the ammonium is going to be assimilated by three different permeases that are under the NCR control. So when you have amino acids, or this group of orange amino acids that are low, especially the glutamine, you're going to have a very early assimilation of the ammonium. On the other side, on the other hand, when you have high amount of ammonium, it's going to down-regulate all the other green amino acids, and it's going to delay their intake. So you always have approximately the same order of consumption, and you can see here on this arrow the general order of assimilation, but the timing, the consumption timing, is very strain and condition dependent. So how does it impact the thiols now? Very important with our topic. Well, the thiols uptake is done through these general carriers, the GAP1P and the OPT1P, that are, you understood, controlled by the NCR. So when you have lower level of nitrogen, especially these orange amino acids and ammonium, the NCR is going to release earlier these two permeases, the GAP1 and OPT1, and you're going to have more thiols uptake into the cell. When you have a lot of ammonium, it's going to be taken preferentially, and it is going to delay the intake of the thiols. So therefore, you're going to have less thiols uptake. The second impact is going to be on the bioconversion of these precursors. So the NCR also downregulates the IRC7 gene. So what it means is, as long as the NCR is active, meaning lots of certain amino acids and ammonium, you're going to have overall a lower release of specifically the 4-MMP. So overall, the conclusion is when you have high level of nitrogen and more specifically high level of ammonium, you're going to have an active NCR, and this is going to result in a decrease of thiols uptake, but also bioconversion. And here you go, Marcia.